students, welcome to part 2 of protein synthesis. Hopefully you've watched the Amoeba Sisters video on protein synthesis, so you have an understanding of what to expect. So in the last video you learned about how DNA is replicated, and that occurs in interphase just before either mitosis or meiosis. But in the second part of this video, or in the second part of this lecture, we're going to learn how DNA codes for proteins. What does DNA actually cause our cells to do? So if we think about it in this respect, I know that the title of the slide says the flow of genetic information from DNA to RNA to protein. If we write it like this, DNA codes for RNA, and RNA then tells the cell or codes for the different proteins that cells make. So DNA is this stored molecule inside of our cells that acts as our instruction booklet. But our cells can't read DNA directly. DNA you can almost consider as like a bunch of symbols as far as the cell is concerned. The cell really doesn't know what to do with these letters of DNA that it can't read. It's almost like hieroglyphics or a language that the cell can't read. So what the cell first needs to do is transcribe or rewrite that DNA information into a form that it can understand, and that form that it can understand is RNA. So the cell needs to first convert DNA to RNA, convert this written language that the cell doesn't understand to a language that it does understand, that it can recognize. So if DNA is unrecognizable, so to speak, that's not a correct analogy, but let's just say for explanatory purposes, let's say that the cell doesn't recognize what DNA is trying to tell it to do. DNA can be rewritten into RNA during this process called transcription. So that's the conversion of a DNA message into an RNA message. So rather than having just a bunch of symbols, for example, let's say that those symbols represent the nucleotides of a DNA molecule. In transcription, we convert those symbols to actual letters that the cell can recognize. And so now the cell goes, oh, I see, I recognize those letters. And I recognize those letters come together to make words and sentences and paragraphs and chapters. That's something that the cell can read. And so now that we've got this molecule of RNA made that acts as a paragraph, for instance, now the cell can read that paragraph in this process called translation and translates that RNA message into a protein. Or another way to say that is the cell can read the RNA message and go, oh, I know exactly what protein to make. And remember, proteins are polymers of amino acids. So from reading this nucleotide sequence right here, the cell knows how to assemble the protein that it's been told to assemble. So I'll just write A's here, and the A's here stand for amino acids. Remember, amino acids come together to make proteins. So from these symbols that the cell couldn't really read directly, didn't really know what to do with, the cell can convert that to letters that it can read, and then it knows which amino acids to put together in which order so that it knows which protein to make. So if we look at this slide right here, at the top it says the DNA genotype is expressed as a protein which provides the molecular basis for phenotypic traits. That's a mouthful. And so really what that means is the DNA genotype, what your genes say you are. Let's say there's a section of DNA that has this nucleotide sequence. So that DNA genotype is ultimately expressed as a protein. Well, that's what we were just learning about, right? That DNA gets converted into RNA, and that RNA then gets converted into a protein. And let's just say, for instance, this protein is lactase, the enzyme that helps you break down the sugar in milk. So this would be a phenotypic trait, some type of protein that you actually make. So just like in DNA replication, transcription and translation have enzymes that are going to help carry out this process. There are players here in this process that we need to become familiar with. You need to know what their function is. You need to know what their name is. So first up, an enzyme called RNA polymerase. This is the enzyme that initiates transcription. Really what RNA polymerase does is it begins reading the DNA. And in protein synthesis, you're not going to read, or RNA polymerase isn't going to read an entire DNA molecule. It's only going to read sections of that molecule, sections on that DNA molecule that we call genes. So RNA polymerase is only going to read a small portion of that DNA molecule, 1, 2, maybe 3 or 4 genes at a time, read that DNA, and then determine which, the cell will determine which protein to make. There's also messenger RNA. This is the RNA molecule that's first made from the DNA. So let's see, let me switch colors here. Let's use blue. Remember that DNA gets converted to RNA. So the first molecule that's produced here in transcription is a molecule of what we call mRNA, messenger RNA. And that's going to code for a specific amino acid sequence. And so mRNA is referred to as messenger RNA because it's carrying a message about which protein the cell has been instructed to make. That mRNA molecule then, like I said, is carrying that information. And from that mRNA molecule, during translation, the cell translates that message and goes, okay, thank you. I know exactly which amino acids to bring and in what order in order to make the protein that I'm being told to make. Where that protein is made is in an organelle called a ribosome. So a few lectures or almost a month ago now, we learned about organelles inside of a cell. Ribosomes are a type of an organelle. Ribosomes, as you recall, are the cellular machinery that builds proteins. This is where amino acids are going to be strung together one after the next to create a protein. It's these ribosomes that are going to read the messenger RNA, and at the same time, based on what that messenger RNA is telling it, is going to bring amino acids together in a specific sequence, string those amino acids together in a sequence in order to create the protein that the mRNA has instructed it to make. Ribosomes are found in the cytoplasm. And so this construction of proteins occurs in the cytoplasm. Whereas, so that construction of proteins is called translation, right? That occurs in the cytoplasm. Transcription, all this reading, occurs in the nucleus. I'll show you that here in a second. And then there's transfer RNA. Transfer RNA is a molecule that is going to capture the amino acids in the cytoplasm one by one and bring them to the ribosome. So it's transferring, not transferring RNA, but it's transferring or bringing amino acids to the ribosome in a very specific sequence based on the message or the sequence of the message in the RNA. So those transfer RNA molecules, they pick up the appropriate amino acid and they bring that appropriate amino acid in the appropriate sequence. Well, transfer RNA does this by reading the mRNA molecule. And it doesn't read the mRNA molecule letter by letter or nucleotide by nucleotide. It reads the sequence in triplets. It reads the mRNA message in triplets. And those triplet bases, are going to be matched up to a location on the tRNA called an anticodon. So a triplet, let's say, has the sequence, the base sequence AUG. Think of that as one word, AUG. So that triplet is also called a codon. So it's a code, it's a word telling the cell which amino acid to bring at the right time. Each tRNA molecule has an anticodon, something that matches the codon, or a base sequence that matches the codon. So this is how the tRNA matches up with that mRNA sequence. This will be a little bit clearer to you as we look at it in pictorial form. Okay, just to kind of sum up what we've just gone over is that DNA is found in the nucleus, right? So here's your molecule of DNA. Transcription occurs in the nucleus. And the molecule that's made from transcription is a molecule of what we call mRNA. And that's what you see here in pink. And that mRNA is going to leave the nucleus and make its way to a ribosome in the cytoplasm. And within the cytoplasm, from that mRNA molecule, I'll just write mRNA, that mRNA molecule inside of a ribosome is going to code for a specific protein. Okay, so let's write that sequence up at the top again. DNA gets read to create a molecule of RNA, and RNA is read by the cell to create a protein, or a piece of a protein that we call polypeptide. A polypeptide is just the beginnings of a protein. So if we look at this section of DNA right here, we're just looking at one strand. This is a strand that's to be read. You could think of this one strand right here as being a single gene, and that single gene is going to code for a protein, for instance, or the beginnings of a protein. So in order to know which protein should be made based on this base sequence right here, the cell needs to turn that DNA sequence into a molecule of RNA. And that's the process called transcription. So if a section of DNA is read, we can just go from left to right, AAA, the appropriate RNA sequence, or the appropriate base pairing for RNA would be U, U, U. Remember that in a molecule of DNA, when we're replicating DNA, A always pairs with T, but when we're making a molecule of RNA, A then pairs with U, or gets converted into a U. And then C and G still pair up like this. Now I say pair up, we're not pairing an RNA molecule with a DNA molecule. What I really mean is that a molecule of RNA is built based on this sequence that you see here. So the cell reads this first base first, and then brings to the growing RNA molecule a uracil. And then it brings another uracil because the DNA next letter was A. And the next letter in the DNA molecule is another A. So we bring to that growing RNA sequence another U. And it reads the DNA sequence, and it sees a cytosine, and the cell then brings a G to the growing RNA chain, and so on. So based on what the DNA sequence is, we make an RNA molecule. In translation, the second process in protein synthesis, the cell now is going to read each one of these bases in threes. So these three bases will be read, U, 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 and then these three bases, and then these three bases, and then these three bases. Those are called triplets or codons. So U, U, U is a codon. What that means is it's going to code for a specific amino acid, saying this specific amino acid goes right here in the first position. And then GGC codes for another amino acid to be brought and put in the second position, and so on. We'll go through this a little bit more in the next slide. So in translation, we get this conversion of an RNA sequence to an amino acid sequence. And remember that amino acids, those are the polymers they are the monomers of proteins. So this would be one amino acid, here would be another, here would be another, and here would be another. Now there are about 20 different amino acids. So each one of these amino acids, based on its codon, might be slightly different. Let's have a look at that. So again, 3 nucleotides come together to make a codon, and each codon specifies one amino acid. So again, the authors are giving you this example, AAA or ACG, CUG, AUU. Those are all codons. There are 61 total possible codons or combinations of this AUCG alphabet that can come together to make 20 different amino acids. So for instance, the codon AUG, we know that that's a codon for an amino acid called methionine. Whenever an enzyme in translation sees methionine, it knows, I should start reading this DNA sequence, this RNA sequence right at this point. And there are 3 total stop codons. So think of AUG as like the capital letter of a sentence that tells you, begin reading the sentence here. And the stop codon being like the period. So that everything in between that capital letter and that start codon is the sequence to be read, the meaning or the bulk of the sentence. So in this picture, you see a chart. It's drawn as a square here. Sometimes you see it drawn as a circle. And what it's meant to do really is to help you determine which amino acid is coded for by a specific base sequence. So in the squares, you see base sequences, for instance, like UUU or UCU. Remember, those are nucleotides, or those are nitrogenous bases. The U standing for uracil. So that would be uracil, uracil, uracil. But those are bases. And remember that that base sequence codes for a specific amino acid. that specific amino acid coded for by UUU is an amino acid with the name phenylalanine. Phenylalanine. There's also another codon, UUC, that also codes for phenylalanine. So there can often be more than one codon that codes for the same amino acid. This one right here, for instance, right below it, let me change my pen color, UUA, uracil adenine, codes for the amino acid leucine. But so does uracil guanine. So you see there's some repetition here. If you'd like to learn how to read this chart, if you have a sequence, let's just say, I'll just make up a random one, G, A, G, that's a codon. Guanine, adenine, guanine. Well, look at the position of the letters. This is the first, second, and third. So the way that you read this chart to determine which amino acid GAG codes for, you read down, you read down this line first. So in our example, G is the first letter. Okay, great. So you're in this bottom row. Now the next base is A. You're going to read across the top to the A column where you get here. But now you still have to go all the way down to the bottom, right? So you're at GA, and then the third base you read down the right side. You go all the way down to the G on the right side, and that brings you to our codon example GAG. And that codes, then, according to this chart, for an amino acid called glutamine. Let's look at another example. Let me erase all of this. There's a worksheet in your supplementary worksheets folder called Transcription and Translation that gives you some practice in converting these codon sequences to amino acids. It also helps you to learn how to convert DNA to RNA sequences. That's part of your homework. That's something that you need to have turned in for Unit 3. So in order to complete that homework assignment, you'll be using this chart, or there's a chart in your textbook. But just so you know, you'll have to use a chart in order to complete that assignment. So let's go ahead and do another example. Let's use another pen color. Let's do A, C, C. First, second, third. So remember, you're going to read the first row. So you go all the way down to the A. And then for the second base, you read across the top. So you're going to go all the way over to the C. And for the third base, you go down, back down the columns. And so in this case, we're going to go down to the C. Right here, and that brings us to ACC. And so we can determine by using this chart that the codon ACC, or adenine, cytosine, codes for an amino acid called threonine. You don't need to know the full names. You can use the abbreviations. That's not important. But it's important that you do understand how to use this chart. Another thing that this chart shows you is that some codons act, remember, as capital letters or periods, meaning it tells the enzymes where to begin reading on that RNA code and where to stop reading. So AUG, for instance, that's a start codon. It codes for an amino acid called methionine. And all of these here in red, those are all stop codons. All of those act as periods to a sentence. UAA, UAG, and UGA, all stop codons. They act as periods in a sentence telling the enzyme, okay, stop reading that RNA sequence right here. U, U, C. So we read first column, and then across the top, and then all the way down the right column. So U, U, and all the way down that right column. I guess it's not all the way down. It's right here. UUC, the next amino acid brought in the sequence, UUC, codes for... Okay, so let's carry out transcription. And in this DNA molecule, the authors of your textbook are telling you, okay, here's the strand to be transcribed or turned into a molecule of RNA. So let's go ahead and match the base pairs based on what the DNA strand says. So T is going to match up with A. A is going to match up with U. Remember, in RNA, the base here is uracil. In DNA, A would match up with T, but we're making a molecule of RNA, so in RNA, A matches up with the base uracil, abbreviated by that word U, or the letter U. This C right here is going to be the same. It's going to match up with this G, and then we can just continue on. So again, another A, a C, and instead of thymine or the T's, we've got U, 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 and then another A and another G. Sorry, that's a little crooked there, but each one of these are codons. So each one of these are a triplet. Remember, AUG is a start codon. So that's like the capital letter in a sentence. It tells the enzyme, okay, start reading here. And then it continues to read all the way to the end of the sentence. UAG is a stop codon. And that tells the enzyme, okay, stop reading the sequence there. So really, everything in between is, that sequence is carrying the information about which protein to make. Alanine. So this is essentially what we just did. And if we were to go back to our chart and find the amino acid encoded for by AUG, we'd find that it is methionine. AG, this next codon, codes for an amino acid called lysine. UUU codes for an amino acid called phenylalanine, and then UAG is a stop codon. And this first one, AUG, is a start codon. And that's exactly what we did in the previous example. So now you've got methionine, lysine, phenylalanine, and then that would be another amino acid here. There is your amino acid sequence. And remember that amino acids are monomers of proteins. So we're gradually building proteins, stringing those amino acids together at the end. Okay, so remember, this is the process of protein synthesis. DNA to RNA to protein. And the first step in this process is transcription. Okay, so transcription is going to begin with what we call initiation. There's an enzyme called RNA polymerase that's going to attach to a very specific location on the RNA called the on the DNA called the promoter. That's like a little flag that tells RNA polymerase, hey, attach right here. This is exactly where you need to start reading. And from then on, RNA polymerase continues down that DNA molecule in a process called elongation, essentially reading all of the words from the promoter and forwards. And so that elongates this RNA molecule. And then the third phase of translation is the RNA polymerase is going to get to a section of the DNA where it recognizes this is exactly where I need to stop reading. And that phase is called termination. That RNA polymerase reaches a sequence of bases in the DNA that tells it or signals the end of a gene. So we'll go back to where we didn't look at that as an actual example. Left off. Let's look at it all at once. It's a little bit easier. Okay, so here's the molecule of DNA, this helical structure that you see here. Here's RNA polymerase. There's RNA polymerase. And here would be the promoter region. This would be a nucleotide sequence that tells the RNA, start attaching here. That's almost like a capital letter. It's not the same thing as a start codon or a stop codon, not necessarily, but it's the point where the RNA molecule, the RNA polymerase, knows to start reading. And look down at the other end. There's the terminator. That's a sequence of DNA bases that tells the RNA polymerase, okay, stop reading here. So that is when that RNA polymerase binds to the promoter. That's called initiation. And then you can see that RNA polymerase is going to read in this direction in the process called elongation. It continues on, and RNA polymerase right there will ultimately end up at the terminator, or that section of DNA that tells it to stop reading. And look what's been made here. This pink ribbon has been elongated. That pink ribbon is RNA. So that process was called elongation. And then when RNA polymerase eventually jumps off the DNA molecule, that's called termination. So those are those three phases of codes for transcription. Now, translation is a little bit trickier to understand, but if you're good with the terminology, if you understand what all of these words mean, then this will be a little bit easier for you. So now that we've got an mRNA sequence or an RNA sequence, we can turn that into an amino acid sequence. And this is how it happens. So let me describe to you first, or remind you about ribosomes. Ribosomes come in two parts. And let me just draw it over on the picture on the right, because I don't think they've done a fantastic job of explaining this to you. Here's the small unit. And here's the large ribosomal subunit. So each one of those brown pieces are parts or units of a ribosome. What will happen is initially this mRNA molecule is going to bind to this small ribosomal subunit. And that RNA molecule, remember, has a sequence. It's got a base sequence to it. For instance, let's just say this next codon is UUC, and then the next one is GGG, just as an example. That small ribosomal subunit is going to feed the mRNA molecule into it. And based on what its codon sequence is, or its nucleotide sequence is, it's going to read these triplets, or those triplets are going to be read. And based on what those triplets are, there's going to be a molecule of transfer RNA, tRNA, that's going to have exactly the opposite codon called the anticodon. So let me go ahead and erase this green line because Can't see anything. So if we look at the codon AUG, a molecule of transfer RNA with exactly the opposite codon or the anticodon, UAC, will recognize it. So a tRNA with UAC as its anticodon binds to the AUG codon on the mRNA. And that UAC transfer RNA is bringing the amino acid methionine. Remember, that's a start codon. It's still an amino acid. It's like a starting amino acid. So this transfer RNA knew, so to speak, to bring that methionine to this starting position because it matched up with the AUG on the mRNA. And then it's going to, there will be another transfer RNA that'll come in right to this spot that will have the anticodon for UUC. So what might that be? What would be the anticodon for UUC? The anticodon for UUC would be AAG. So there's going to be a tRNA molecule out there in the cytoplasm that has that AAG codon. And it's going to be carrying an amino acid as well. Which amino acid? Well, we can go back to our chart and let's look and see what UUC is where we left off. So this amino acid right here being brought by that tRNA will be phenylalanine. What will happen next is there's going to be a second part to the ribosome called the large ribosomal subunit. That's going to bind, so this structure binds to the tRNA, and that creates what we call P and A sites. So here's the P site, here's the A site. All that means is that A site is where the next transfer RNA is going to bind. This tRNA right here will pop off and go back into the cytoplasm, and it will leave this methionine or this amino acid behind on top of this next tRNA. So this tRNA, the first tRNA, is going to leave and move out of the way. Oops. So let's look at this a little larger. This is a larger picture for us to see everything that's happening here. Here's the tRNA right here. It's going to pick up that amino acid, and with a little bit of ATP, that amino acid is going to bind to the tRNA, and that amino acid is going to then be brought by the transfer RNA to that A site right there. This transfer RNA will leave, leaving the methionine behind attached to the new tRNA. Oops, sorry. The new tRNA that will bind right here. So if we look at it all together, you get this gradual growing polypeptide chain by one tRNA coming in, dropping off its polypeptides and then leaving. So it's going to transfer this growing polypeptide chain to the new amino acid, and then this transfer RNA leaves. And eventually, all of that growing polypeptide will pop off of the transfer RNA when the ribosome gets to this point of termination, that RNA sequence that says, okay, stop adding amino acids. So here's just a really nice picture that shows you everything all at once from transcription, and then all the way through the process of translation, where the amino acid attaches, polypeptide synthesis begins, the putting together in a string all of those amino acids. That's called elongation, and then termination, where that polypeptide pops off of the very last transfer RNA, and that polypeptide then remains in the cytoplasm. So anywhere throughout the process of either DNA replication or within protein synthesis, within transcription or translation, a mistake can be made, or a mistake could be made to a growing polypeptide chain, or a mistake could be made in adding an inappropriate nucleotide somewhere, or even to the original DNA molecule. There might be a change in the nucleotide sequence, meaning there may be a nucleotide somewhere in the DNA or RNA sequence that doesn't belong, and this is called a mutation. And mutations can be very small. They could just be one nucleotide pair. So let's say, for instance, just one letter in a word could be off. Or you might have large chromosomal regions. This would be the equivalent to like several sentences being off or incorrect in a paragraph. And then that changes the entire meaning of the paragraph, for instance. So that's what mutations are. And they can really be divided into two general categories. One category is called base substitutions, and the other category is called insertions or deletions. Let's learn about base substitutions first. And what those do is they replace one of the nucleotides or one of the nucleotide bases with some other one that doesn't belong. This is a letter replacement. This is inserting the letter A, perhaps where the appropriate letter would be E or O. So this isn't always bad. Sometimes this has no effect at all. And this makes what we call a silent mutation, meaning just because the word is one letter off, it can still be understood. I'll show you an example in a second. It sometimes, though, can change the amino acid coding, producing what we call a missense mutation, which actually causes a different amino acid to be brought to the growing polypeptide chain and cause a condition like epidermolysis bullosa. That's just a really fancy word for a condition where the outer layer of the skin is very thin and peels off and blisters. This is obviously a genetic mutation. So sometimes those mutations can have no effect on the organism at all. Sometimes they can have huge effects. It just totally depends on where that is and a number of other factors. A base substitution could produce an improved protein that enhances the success of an organism, or a change in an amino acid could create a stop codon, producing what we call a nonsense mutation, one like what we call a type of muscular dystrophy called Duchenne muscular dystrophy. This causes severe muscle degeneration in individuals. So you can see the range of complications that base substitutions can have. Let's look at the next type of mutations that can occur, deletions or insertions. So this is exactly what these terms imply, is that a base could be deleted or an extra one can be inserted. And this causes in what we call a reading frame shift, where the sentence is no longer read in the appropriate sequence. I'll show you an example. Let's look. Here we go. All right, so a frame shift mutation I'll get to at the very end. Let's look at this normal sentence up here on top. And as you read it, it says, DNA has all you can ask for. Well, that should be the normal DNA sequence. If we replace if we replace the U in the word U with an O, well, a sentence still sounds very similar. It can still be read as DNA has all you can ask for. That's an example of a silent mutation. Doesn't change the meaning of the sentence at all. So while it's a mutation, it's a change in the nucleotide sequence, doesn't have any effect on the organism. If, for instance, the L in the word U is substituted for the Y, then the sentence reads, DNA has all Lou can ask for. Well, that doesn't make any sense. That kind of almost changes the meaning of the sentence. So missense mutations can be very harmful. In a nonsense mutation, as in this example down here in number 4, right here, this should be a complete sentence. This sentence down here should say DNA has all you can ask for. But perhaps AUG was or not AUG, UUG I think is the stop codon. Let's say there's a sequence in there that tells the RNA to stop being read. And so now you have half of a sentence. And that's nonsense. It doesn't say anything. That can cause severe issues. And then lastly, here's a frame shift mutation. So let's say, for instance, that one of the nucleotide bases is deleted. Look, you don't have as many letters here. We know that this RNA sequence has to be read in triplets, right? So these three are read together. So far, DNA makes sense. HAS, so far, makes sense. But in this third word right here, this sequence, only 2 of the bases are the same, and the L has been changed to a Y. And another way to say that is that there was a different base here. It was that L was right here. But now, everything has shifted to the left. So the Y in U is now being read with the AL, the first 2 AL letters. And so now everything is shifted. And so it shifts the reading frame. So now the sentence doesn't make sense either. Here are a couple of examples of a pretty common mutation. Maybe some of you guys even have this condition as a result of a mutation. This is a mutation called, or not the mutation, but it's a condition called heterochromia. Heterochromia means different colors. And there are different versions of it or different forms of it. You can have complete, sectoral, or central heterochromia. So Mila Kunis right here has one totally different colored eye than the other, whereas this one over here is more of a hazel color. This is more of a greenish blue. That's complete heterochromia, meaning completely a different color. Whereas I think this is Kate Bosworth. If you look at her eyes, she's got sectoral heterochromia, where one eye is completely blue, the other eye is blue on top, and brown on the bottom. And then Angelina Jolie has what we call central heterochromia, where this might be a little bit more difficult for you to see, but the area right around the pupil is more of a a yellowish color, whereas the area a little further around the perimeter is more of a bluish color. That's called central heterochromia. There's another type of deletion mutation called cru de chat, but you guys don't need to know about that. That's something I would normally explain if we were in class. So the word for the production of mutations is called mutagenesis. Mutagenesis can occur in a number of ways and can be caused by sometimes just spontaneous errors that occur during DNA replication or recombination during meiosis, For instance, anything that in the external environment that causes a mutation is what we refer to as a mutagen. So high energy radiation, like X-rays, can cause mutations to DNA. Ultraviolet light, the sun, as we know, can cause mutation to DNA. But also exposure to certain chemicals can cause a mutation of the DNA. So all of those things would be referred to as mutagens. So that's the end of the protein synthesis lecture. I know that Amoeba Sisters should have a video on mutations you can also watch, but it's not in our Canvas folder. But if you wanted to learn more about mutations, you could just do a Google or a YouTube search and find some more videos on that.